sun's hitting my face. I like this ray. Ray, a drop of golden sun. Alright, I'm done tweaking. I gotta re-sign into all this. Damn. I gotta re-sign in. Hello. Am I? Okay. We're chilling. Okay. Let's do it. Let's do it. Let's do it. Okay.
Okay, so this is the second video in the discussion about gene expression. How do we go from information stored in DNA and how do we make proteins? So a general cartoon and outline of how this occurs in eukaryotic cells. We know this is eukaryote, obviously, with DNA enclosed within a membrane-bound structure called the nucleus. So we start with transcription, all right, and with transcription, we're going to use DNA as a template to make messenger RNA. But we first make it as a transcription. Part of this word is a scribe. And historically, scribes for the communication in the world language, from a language of nucleotides in the form of RNA, all right, with a nucleotide polymer. And we're going to use that information to make proteins, which are a polymer of amino acids. So if you think about translation, you think about it as a process of going and using the nucleotide sequence, the RNA nucleotide sequence, to create a protein amino acid sequence. When we talk about translation, this is just a kind of general scheme of what's going on. I'll kind of introduce you to different parts, and then we'll break it down step by step. All right. First off, we have the ribosome, which is our molecular machine. All right. It's going to be consisting of proteins and nucleic acids. There's going to be a large subunit up here and a small subunit down here. All right. We're also showing you in red, and this will be consistent throughout this whole video. All right. And all these PowerPoints is the reds are messenger RNA. All right, you can see that there's a 5' end and a 3' end. We'll learn that the messenger RNA is actually read from 5' to 3'. We're also going to see another kind of RNA that we haven't talked about yet right here, being a transfer RNA. And I'll talk a little bit more about transfer RNA, but the transfer RNA's job is to bring in the proper amino acid to the ribosome. So which amino acid goes next? Well, that tRNA is going to help us figure that out. All right. So first thing we need to understand is how do we read messenger RNA? Remember, messenger RNA is going to contain these U's and G's and U's and C's and A's, all these different nucleotides in a very specific order. All right. And that order is going to be dependent upon whatever the order of the sequence was in the DNA, right? So if you go and you connect, you can see that there's a G for a G, a T for U. Remember, uracil only in RNA, so they don't have a T. But you can see that there's a T, a U, a T, because these correspond to one another. All right, so the sequence that was in the DNA, that sequence is represented in the RNA. All right, so when we go ahead and read our messenger RNA here from 5' to 3', we're going to read it based on what we call a codon. A codon is a triplet of nucleotides, 3 nucleotides in a row. So you can see here that the first codon will be UGG. That's codon number 1. Codon number 2 is UUU. That's codon number 2. Codon number 3 is GGC. You get the idea that we're going to read these nucleotides 3 at a time. And I'll show you how we do that in the ribosome. But the first important thing that you get is that we don't just read the messenger RNA sequence every nucleotide. We read them as triplets that we call codons. Now, the reason that this is important is because each codon is going to code for a specific amino acid. In this case, you can see that the first codon, UGG, codes for the amino acid tryptophan. The next one, UUU, codes for the amino acid phenylalanine. GGC codes for the amino acid glycine. Now, I'm not going to ask that you know which are which. You don't have to know that. Okay, but you do need to know that each codon is going to be specific for a particular amino acid. These are all the different codons. Again, do not memorize these. What you need to know is that for the 20 main amino acids, so there's 20 main amino acids, all right? Because we end up breeding things in triplets, there is a combination of 64 different codons that we can have, because the first position can be an A, a U, a C, or a G. The next position can also be a U, a C, an A, or a G. And that third one can be each of the 4. So when you get the different combinations, AAA, AAU, AAT, what have you, you have a lot of more combinations than you would have for 20 different amino acids. Well, this is because some amino acids have multiple codons that code for them. So, for example, you can see over here at the top right-hand corner, we have CYS. This is cysteine. Both UGU and UGC, both those codons would bring in the same exact amino acid into the sequence. All right. There are a couple codons that you do need to know. One codon that you do need to know the sequence of is down here towards the bottom left, and that's AUG. AUG is our start codon. This is the one that starts every single protein. It codes for the amino acid methionine. It's abbreviated MET. I do want you to know that. I want you to know that AUG is the start codon, that it codes for the amino acid methionine. What you also need to know is that there are 3 different stop codons. You can see them up here at the top right. I don't need you to know the sequence name. I don't need you to know UAA, UAG, and UGA. Don't care. Just know that there are 3 different stop codons. Now, the next thing you need to understand about codons is because we read them in triplets, it matters very much where you start reading. Okay, so if you look here, they're giving you 3 different scenarios of all the same exact sequence. Take a look first at the sequences. Okay, it all starts C's, U's, C, A. These are the G. I did it off. G. So these are the same exact sequences. All right, let me try to get the eraser. So these are all 3 of these happen to be the same exact sequence. In number 1, they start you on the first C here. So by reading triplets, it starts C, U, C.
I do see.
E, C, U, G, U, U, okay, and so forth. Now in the second one, what we call a reading frame is shifted. The reading frame is basically where you start. All right. So if you look here, since they have this being
Okay. Damn, I got so much fucking shit.
being the first codon, we have UCA, then GCG, UAA. Basically, what you end up doing is you can have the same exact sequence, but if you have a different reading frame, you come up with a different protein. So, for example, in number 1, the amino acids go leucine, serine, valine, threonine. But in the second sequence of amino acids, using the same exact RNA sequence, we have serine, alanine, leucine, and proline. And those are completely different. And remember back to the last exam, all right, 2 exams ago. When we talk about proteins, their amino acid sequence is the most important aspect of it. Every protein has a specific amino acid sequence. So if you don't read in the proper reading frame, you completely change the amino acid sequence, which means you completely change the protein. Bottom line is, every messenger RNA has to start in the right exact place so that you can be in the right reading frame. Now, before I show just tell you, the way that we do it, and I'll show you this in a bit, the way we make sure that we start in the same reading frame every time is we always start on the start codon, AUG. As long as we start on that start codon, and all proteins will start on just the start codon. Remember, there's only one of them. AUG is the only sequence, is the only codon, I should say, that codes for the start of amino acid protein synthesis. It codes for methionine every time. So, how do we know which amino acid to bring in to the ribosome based on the codons? Okay, so we know that there are codons. All right, we can see here that there's the 3 codons. All right, how do we know which amino acid is brought in is based on a structure called a transfer RNA. All right, the T of tRNA stands for transfer. This is an RNA molecule, so it's single-stranded. You see that there's a 5' end and a 3' end. Even though this is single-stranded, it actually folds and bond base pairs with itself. So you can see what they're saying, hydrogen bonds right here. That's because the A's and the U's and the G's and the C's are hydrogen bonding with one another, much like how the anti-parallel two strands of DNA are held together. Here, still hydrogen bonds between base pairs of C's, base pairing with G's, and A's, base pairing with U's. Now, this is important because this folding and interaction allows for a very specific three-dimensional structure that has two important sides to it. The first side here is this 3' side that I've already shown you. That's where we attach our amino acid. All right, one of our 20 amino acids will be attached here. Which one is specific for the tRNA? Every tRNA has a specific amino acid that it carries. The other side is the opposite side, and that's called the anticodon. The anticodon is just like the codon in that it's a triplet of nucleotides. But what this triplet of nucleotides will be is that it will be specific for a codon. So what we're going to see is that there's going to be a codon anticodon pairing to make this process work.
Hence the name anticodon. Yep.
Now, to make sure that we have the proper amino acid hooked to the amino acid based on the codons, okay? So we know that there are codons, all right? We can see here that there's the 3 codons, all right? How do we know which amino acid is brought in is based on a structure called a transfer RNA, all right? The T of tRNA stands for transfer. This is an RNA molecule, so it's single-stranded. You see that there's a 5' end and a 3' end. Even though this is single-stranded, it actually folds and bond base pairs with itself. So you can see what they're saying, hydrogen bonds right here. That's because the A's and the U's and the G's and the C's are hydrogen bonding with one another, much like how the anti-parallel two strands of DNA are held together. Here, still hydrogen bonds between base pairs of C's, base pairing with G's, and A's, base pairing with U's. Now, this is important because this folding and interaction allows for a very specific three-dimensional structure that has two important sides to it. The first side here is this 3' side that I've already shown you. That's where we attach our amino acid. All right, one of our 20 amino acids will be attached here. Which one is specific for the tRNA? Every tRNA has a specific amino acid that it carries. The other side is the opposite side, and that's called the anticodon. The anticodon is just like the codon in that it's a triplet of nucleotides. But what this triplet of nucleotides will be is that it will be specific for a codon. So what we're going to see is that there's going to be a codon anticodon pairing to make this process work.
I don't remember this shit at all.
Now, to make sure that we have the proper amino acid hooked to the correct tRNA, we have an enzyme that does it. These are called the amino-acyl tRNA synthetases. Break it down. It's not a real big tough word. Okay. If we start with amino-acyl, okay, think of our amino acid. All right. Think of tRNA. That's our carrying nucleic acid. And then synthetase, synthesize. So this enzyme links together, synthesizes an amino acid getting linked to a tRNA. All right. So you can see that the reason it does this is because there's a very specific three-dimensional shape to the enzyme. All right, they show you the kind of lock and key model here in the middle. Down in the bottom right, they actually give you
Okay,
a
I'm
computer
lost. I'm fucking
simulator.
lost. I'm fucking lost. What? What?
Molecular diagram showing you a lot of the atoms that are involved in this. And you can see here that the tRNA, all right, look right here for the tRNA. This tRNA is specifically bound into the enzyme in a way so that this end over here, all right, the end right here, that end can have the right amino acid being brought in to link to the proper tRNA. Now, to form this bond between the amino acid and the tRNA, we have to burn some ATP to do this. We're forming a covalent bond. We've talked about that before. Bonds are energy. So we use an ATP molecule to form that bond between the amino acid and the tRNA. So now looking at a ribosome as our molecular machine that does translation. Again, I talked about the fact that there's a large subunit and a small subunit. You can see here in the middle that there are three different sites that they say are important: the E site, the P site, and the A site. The A site, I'll show you again in a minute, is the amino-acyl site. This is where a new tRNA comes in. The P site we'll call the peptidyl site. All right, this is where the growing polypeptide chain, the growing protein comes from, is attached to. And then the E site, or we'll call the exit site. And this is where an old tRNA that's lost its amino acid coming in is going to be found. You can see here the mRNA down at the bottom, going from 5' to 3'. It'll be read in this direction. All right, so this part down here at the the 5' end, that part's already been used to make it. So these parts, these codons right here, would have coded for these amino acids up here. This is our growing polypeptide chain coming out of the ribosome. So out of the large ribosomal subunit is actually a tunnel. It's a physical space that this growing polypeptide chain can grow out of and leave the ribosome. Now, another scheme of it to go over this a little bit more cartoon form. Again, the most important part of this is that we have the two ribosomal subunits coming in to form these three important sites here in the middle. Okay. The A site is where the amino-acyl tRNA is going to come in. the new one carrying a new amino acid. We have our P site, the peptidyl tRNA site. This is where our growing polypeptide chain will be coming out of this, all right, exit tunnel.
Is this even part of like my... I need to learn this. No.
And then over here, our exit site. This is where our old tRNA can fall off and go out because its amino acid was already added. Its amino acid's now part of that growing protein chain. So remember I talked about transcription in the last video, and I said there's 3 steps: initiation first, elongation, and then finally termination. Translation is going to be the same exact way. We break it into 3 steps: initiation, elongation, and termination. Now the way that this works is this. You start with only the small ribosomal subunit over here in the bottom left. Small subunit grabs onto into a messenger RNA that is found in the cytoplasm. And it will read along that messenger RNA until it comes to the start codon. Remember that start codon is AUG. Every protein starts with methionine because the messenger RNA's AUG, the start codon, is how we get into our proper reading frame. So when the small subunit gets to the start codon, it brings in what we call the initiator tRNA. This is our tRNA who carries a methionine as the amino acid. And the reason why the tRNA matches is because of this right here. This is our codon-anti-codon binding. So the codon would be AUG in the tRNA, and the anti-codon would be UAC here in the transfer RNA. So again, A's and U's and C's and G's match with one another. So because the start codon is AUG, the tRNA that's brought in will be the one that starts this process.
What's tRNA?
Termination. Translation is going to be the same exact way. We break it into 3 steps: initiation, elongation, and termination. Now the way that this works is this. You start with only the small ribosomal subunit over here in the bottom left. Small subunit grabs onto into a messenger RNA that is found in the cytoplasm. And it will read along that messenger RNA until it comes to the start codon. Remember that start codon is AUG. Every protein starts with methionine because the messenger RNA's AUG, the start codon, is how we get into our proper reading frame. So when the small subunit gets to the start codon, it brings in what we call the initiator tRNA. This is our tRNA who carries a methionine as the amino acid. And the reason why the tRNA matches is because of this right here. This is our codon-anti-codon binding. So the codon would be AUG in the tRNA, and the anti-codon would be UAC here in the transfer RNA. So again, A's and U's and C's and G's match with one another. So because the start codon is AUG, the tRNA that's brought in will be the one that starts this process with the first methionine. Now, by using a little bit of energy of ATP, the large ribosomal subunit comes in, and now we form those three sites. Our E site behind here, this is really our P site, and our A site. And now we have initiated translation. Next step's elongation. Okay, so let's start up at the very top, like we're starting up here at 12 o'clock. Okay, and look at this ribosome making this growing polypeptide chain. You can see that at this point we have 1, 2, 3, 4 different amino acids linked to this growing polypeptide chain. The last amino acid, number 4 right here, is still linked to the last tRNA that came in. All right. So we have the tRNA with the growing polypeptide chain here in the P site. All right. And in the
What is tRNA?
A site is going to be our next codon. Okay, so I just circled. In that A site is the next triplet, our next codon on the messenger RNA. That codon will be specific for this anticodon on this tRNA, which will bring that in, all right? And when that's brought in, using the energy from GTP, you can see here that this growing polypeptide chain is added to the next new amino acid. All right, so it falls off of this tRNA, and the growing polypeptide in the red arrow here gets added to the amino acid, still hooked onto the tRNA here in the A site. Next step, we go here to the bottom. We're at like 6 o'clock, and we can see where that has been completely added. Okay, so now that process is over. Using the energy from GTP, we shift the entire sequence down. The message you can see here over at 9 o'clock. The messenger RNA shifts position so that the old tRNA gets to leave through the exit site. The tRNA that just got added to, this was the amino acid that just got added to, that's now found in the P site. And now our A site is open, exposing the brand new codon for the next tRNA and the next amino acid to come in. And then we start all over again. So every time we add a new amino acid and shift down, we burn 2 ATPs. I'm sorry, 2 GTPs, my apologies. So we're going to burn 2 GTPs for every cycle that this goes through. This is one of the reasons why we don't want to just use a protein like an enzyme once and make it and just throw it away and make a new one. All right, enzymes as molecular machines that do biochemical reactions, those proteins are very energetically costly to be able to make. Because every time you're going to add a new amino acid, your whole protein can contain 500 amino acids. That's a very energetic process. So we get to translation termination. Translation termination is going to happen because when we get the stop codon, we'll start over here on the left-hand side, our stop codon, one of the 3 different ones, when that is in the A site, a tRNA does not come in. Something else, another protein that's called a release factor. factor is brought in. All right, again, this is not a tRNA. A release factor is going to come in when the stop codon is there in the A site. This is still the A site. Okay, and what the release factor does is the release factor basically causes everything to fall apart. And we use a little bit of energy here by burning 2 GTPs. So the release factor, along with the energy from GTP, causes the whole thing to fall apart, and that releases the polypeptide. All right, if this is just in the cytoplasm, the polypeptide is now folded in the cytoplasm. If it was maybe made into the ER lumen, that's where it would go. All right, if it's going to be like a secretory protein. So, starts with initiation, goes through elongation, ends with termination. This is kind of a whole scheme of what we talked about so far, starting up here with transcription in the nucleus up at the top, using the DNA as a template and RNA polymerase as our enzyme to make our messenger RNA, which has to go through a maturation process of adding the 5' cap, the 3' poly A tail, and cutting the introns out and putting the exons back together using the spliceosome. That mature messenger RNA can now leave the nucleus via the nuclear pore complex, where it can encounter the small ribosomal subunit, which will read along to the first AUG, the start codon, which will bring in the large ribosomal subunit, and we can get the process of translation. Here they're showing you one of the steps of translation elongation. This is also showing you that in all of the cytoplasm are all of the amino acids being hooked to their proper tRNAs by the enzymes, the aminoacyl tRNA synthetases. So, that's it in a nutshell for translation. Except for the fact that we have that really kind of pictures talks about how that would happen if it was a ribosome free in the cytoplasm. What this is describing here is how do we get a protein made at the endoplasmic reticulum. So this is kind of busy, but it's numbered, and we'll start just with number 1. Protein synthesis always starts with free ribosomes in the cytoplasm. And when a ribosome starts making a protein, and you can see here, take a look at the ribosome and see the protein. You can see the purple part up here in the exit tunnel still. But you can see that some of the protein starting to come out, and they colored them there in the green spheres because they wanted them to make it look different. And the reason they wanted to make it look different is because if you're a protein that needs to go to the endoplasmic reticulum, the first amino acids that are made are referred to as a signal peptide, because those amino acids, their characteristic allows for the targeting of the ribosome to the ER. Think of the signal peptide like a zip code that you would put on a letter. If you put a zip code on a letter, the 5 digits, and if you include the 4 digits after, or if you do the whole 9-digit, that can get to a specific address. You don't have to write a name or a street number or anything like that. You give, 13126 dash, whatever, 4 more digits. You can get that letter where you want to go. The sequence of amino acids in our signal peptide, those first amino acids in a protein that must go to the ER, that's called the signal peptide, because what it does is that amino acid sequence in the signal peptide binds to a molecule called SRP. SRP is a protein in the cytoplasm. It's called the signal recognition particle. And what SRP does is SRP binds to the signal sequence. That's what it says in number 2. SRP binds to the signal sequence or the signal peptide. And what it does is it causes a translational pause. That's not written here, so make a note of that. It's an important part of it. SRP binding to the signal peptide causes translation to pause. It stops momentarily. And it stops momentarily because we don't want to make the protein until we get to the ER. The protein's supposed to be made into the ER lumen in this diagram. So we don't want to make the protein in the cytoplasm, so SRP stops it. That's one job of SRP, pausing translation. The second part is that the SRP actually has an SRP receptor found in the membrane of the endoplasmic reticulum. So here you can see kind of at the bottom left, it says SRP receptor protein. Here it is embedded in the membrane of the ER. So SRP binds to SRP receptor to bring the whole ribosome complex to the ER surface. At that point, you could see in number 4, SRP detaches and the polypeptide synthesis resumes, so transcription resumes. And now the protein is transcribed, is synthesized into the ER lumen. Number 5 shows you that that signal sequence, that signal peptide is clipped off. And now you have the mature protein found in the ER lumen. So this is how proteins that need to get to the ER do that and are made in the ER. If this were a protein that we needed to be made in the cytoplasm, that protein would not have a signal peptide because that signal peptide is not found on the proteins that are cytoplasmic. That means the protein will just be made in the cytoplasm. The SRP will never interact and it will never have any of these other subsequent events take place. All right, I put a video on.
Yeah, I stopped paying attention like right here. I started feeling like, damn, I'm hungry. Like right here. I saw this shit and I was like, what the fuck? So I didn't really understand the last 15 minutes of that video. I didn't really fully clock it. Clock it. Yeah. Man, why is this the only shit I get in my chat, bro? Why is this the only shit I be getting, boy? Yo, I just saw my own self scratch my Nah. Whenever this happens, my computer starts glitching. What the fuck? My shit like, hello? Why my shit look like that? I love Nick 830. I love Nick 830.
We got to do the power hour and get some amazing wins. So hopefully you guys all enjoyed all that. I'll talk to you guys on Tuesday. Tuesday is going to be the next stream, guys.
Oh.
So I'll see you guys on Tuesday. I'm going to be updating the VOD channel this weekend tomorrow with fresh new content, but there's still some amazing new stuff from this season that's new.
Tomorrow? Yeah, you're Canadian. You're Canadian. Nice try. Burn him. Burn all Canadians. Okay. This is fascinating to me, but I don't understand the end of it. Oh, we still got more shit. But it's whatever. That looks like a photo he took himself. I'm just hungry as fuck. I'm not doing that shit today. Maybe tomorrow. Is this part of the fucking thing? No. Okay, synaptic signaling makes no sense to me. This is a pretty short video. I'm just looking at that like right before the exam. Damn, my shit ain't even in the frame.